as a professor at Cold Spring Harbor Laboratory, I'm really lucky to work on this gorgeous campus here. And if you haven't been to Cold Spring Harbor, please come and pay us a visit. We'll show you around the campus. It's quite amazing. But the most important thing that has been fantastic for me as a professor at Cold Spring Harbor is to head the really expert, exceptional there's scientists from around the world, individuals. You just met Jan, lots of other people here, you can meet other members of my team, and to work with them and really to see their passion for science. And we study basic cancer discoveries, but we want to bring them also to the clinic. So basic mechanisms of science. And so we're focused on cancer. My laboratory is right here. You actually can't see it. There's so many trees in that campus behind where Walter is. And we study cancer genes, and we, of course, want to know, we study various cancers of the skin. Also, we study brain cancers. And we really want to know the genes that are driving these. So as you probably know, cancer is a genetic disease. And for example, there are hereditary forms of cancer. So for breast cancer, for example, there are genes, for example, BRCA1 and 2, the BRCA genes, that can be passed down to a woman, and it gives them a predisposition, a likelihood that they will develop breast cancer. It does not mean they will develop breast cancer, but they're much more likely to do so than a woman who does not have the BRCA mutations. So these hereditary cancers, the familial cancers that are passed down from these unusual versions of the genes, those are kind of easier for us geneticists to study because, of course, you can see like the grandmother might have had breast cancer, pass it down to the mom and to the daughter. So it's easier to get at those cosmic genes that are the culprits. However, hereditary cancers are only about 10% of all cancer cases. The majority, about 90%, are what we call spontaneous cancers. Something goes wrong with a gene kind of sporadically in some cell of the body, and that precludes the cancer. So those two kinds of cases, those are much harder to study. So we are very interested in studying, also we study the genes, as I said. There are two flavors of genes that are really important in cancer. The one flavor are called oncogenes. They're promoting cancer. They can be initiators of cancer, or sometimes those genes that are really important in maintaining the malignant cancer. And those, of course, are the negative genes, but luckily we're all normally endowed with what we call tumor suppressor genes. So these are the natural ability that we normally have within each of our cells that prevents cancer. And so you can imagine that these oncogenes that cause cancer or promote cancer, those that inhibit it, the tumor suppressor genes, are kind of at battle. And so the interactions between those are really important in both cancer predisposition, how it develops, and also how a patient responds to treatment. So the genes are very, very important, but it's not just the genes. The genes are on chromosomes, there's DNA in there, of course, but half of the chromosome, they're actually these other proteins, and it's important how the DNA is wrapped. So these proteins are wrapping very intricately, very importantly, wrapping the DNA into a structure. This is a whole field of epigenetics. So it's not just the genes that one inherits or get mutated spontaneously, it's actually how the DNA is packaged. So that field I'm really interested in as well. So one example that I wanted to talk about of some of my team's discoveries are shown in this slide. It's a brain cancer called glioblastoma. I'll also refer to it as GBM. It is the most common adult primary brain tumor. And what happens, shown here by an MRI, is here is the skull. The patient's actually lying down on the bed. And this is the skull shown here in the white circle. And then the normal brain tissue is shown in this gray matter, this stuff here in the ventricles. But what you can see in this patient is there's this large lesion, and this is the glioblastoma tumor growing. And what I think you can appreciate from this MRI is this is overtaking this normal brain tissue. It's actually squishing the normal brain out of the way and disrupting brain function. So these patients, all of a sudden, a highly functional individual can just have seizures, lose their ability to speak. They have total personality changes. Different things happen depending on what portion of the brain is affected. And about half of the patients who are diagnosed only make it for less than a year. The 5-year rate is really dismal. 5% of people make it. There are about 80 people in this room, so if each of us were all of a sudden diagnosed with glioblastoma, in 5 years, what's 4 people, 4 of us would be still here. So it's really, really dire. And because of this really nasty ability to disrupt the normal brain function, the treatment that people get are surgery because it's squishing the brain, you can't survive. And so my colleague John, who I'll get back to him a little bit later, gave me this image of the tumor he dissected out from one of his patients that he diagnosed this way. And you have to remove that tumor. And then there's repeated rounds for these patients. They undergo repeated rounds of radiotherapy, chemotherapy, round and round again. And you can imagine that's really quite devastating. The tumors, actually, after the surgery, debulking the tumor, and then some of these treatments, everything looks great. People are happy. They're feeling better. They're not having the seizures. The tumor's out of there. However, they seed out, and so a lot of tumors come back actually with a vengeance. This treatment, we call it the standard of care treatment, this surgery and radiation and chemo, it hasn't changed in like 5 decades. It's really changed very, very little. So I'm a geneticist, I'm a gene hunter, and my team helps me do all that. And what puzzled me when I started thinking about glioblastoma is compared to other solid human tumors, there are very few mutations. And this was really weird. So for example, I mentioned that our normal ability to prevent cancer, the tumor suppressor genes, the most famous tumor suppressor gene, I'll argue, is called P53. And almost all human solid tumors have to take out P53. They wipe out our ability to, so they mutate it, for example, or they inactivate it in some way. But when we started looking at glioblastoma, this was not the case. Almost 3/4 of the patients, 71% to be exact, have unmutated P53. So this to me was really, really odd. Like, how could it even be cancer? And I think the most malignant cancer, worse than even pancreatic cancer, if the ability to prevent cancer was really intact, like it appeared to be. So we took a deeper look, and we found that in fact, even though P53, our normal ability to prevent cancer, was not mutated, it still was defunct. It couldn't do its job. So the question was how? So we reasoned, few mutations. We reasoned that there might be a way, if we thought about how the DNA was packaged, the epigenetic machinery, there might be a way where we could target that and essentially find an Achilles heel that would wipe out the glioblastoma. So that was how the project started. Very long project. You can ask me how long later. Basically, we wanted to find something where we could target a piece of the chromatin remodeling machinery and wipe out glioblastoma. And we did this, and we found a completely new finding that there was this pro-cancer gene that was maintaining glioblastoma malignancy. It was vital, and what it was doing is wiping out our normal ability to inhibit cancer by taking out P53, just basically sabotaging it. And so the nuts and bolts shown schematically here are BRD8, that's the chromatin machinery here that is important, and that's maintaining cancer. And it's working with its partners. We did a lot of studies to look at the mechanistic, basically the nuts and bolts of how this was all working. We found its partner, there's a big complex, there's about 18 different proteins there. The details don't really matter. But what BRD8 was doing is using a special motif, a portion of its protein, its heart, I call it, and it's maintaining H2AZ, this protein called H2AZ, at P53 targets. So it's crunching up chromatin, it's making it too compact, and so P53 is not able to get in there and bind and turn on the tumor suppressive power that we all should have. This was a completely new way that we found that BRD8 was working to basically wipe out our ability to prevent cancer. So all of this, it was fantastic for the cancers. And we found, when we looked at hundreds, I think 400 human patient samples, which I'm not showing you here, we found very high levels of BRD8 and this protein called H2AZ. So this is all great for the tumor. It's proliferating madly, it's highly malignant, and it's going and going and going, and it doesn't set on the brakes. However, what we found when we studied this mechanism and all the details of the nuts and bolts is we found a way where we could target BRD8, and specifically by targeting the bromodomain and disrupt this, everything popped off. BRD8 was gone, all these partners magically came off. And then P53, this tumor suppressive power that we had, then could go in and bind its targets, specifically those genes that are important in controlling cell cycle and inhibiting tumorigenesis. And so we think of this as a BRD8 P53 epigenetic switch. If we target BRD8, everything normalizes. Everything from gene expression that we can talk about later to cell cycle when we grow these in a dish, everything is normal, they stop proliferating. And even when we implant these human cells from patients into the brains of mice, we basically use the mice as it's like a vessel where we can study in a real brain, a host mouse, grow the tumors. But when we target BRD8, this inhibits the gliomas from forming, and also importantly extends the lifespan of those mice that harbor this. So here we were super excited. You can't see it down here, but Shireen Sun, a woman who she led this study in my lab when she was a postdoc. We published this in Nature. So we were really happy. We have a lot of scientists in the room. I think I see some, and everybody who's a scientist knows you make one discovery, you're like, yay, but there's 200 more questions. So we at Cold Spring Harbor are a basic cancer center. So we do fundamental research, basic cancer center sponsored by the National Cancer Institute. But that means we're not seeing patients ourselves. We're not treating patients on that campus that I showed you. So what we did is we had this alliance, we made this alliance between Cold Spring Harbor down here, us basic scientists working not just on cancer, but other things. We teamed up with clinicians at Northwell Health, treating various cancers and other elements. So now we have us basic people in the room with clinicians. Here we show the team. I got together with our group, and here we're having a one-day retreat, basically a think tank between us basic scientists at Cold Spring Harbor. Some of my team is here. Here's Gia, and here's Jan. Back here, here are our collaborators here that I'll mention in a minute, who are helping us make compounds. John Moses is leading that team. And then importantly, from the Northwell Health side, these are clinicians, brain surgeons, the ones that took out those tumors. That was actually John Bukvar shown here. Michael Scholder was also helping us with a lot of tumors. And so everybody getting in having the day where we spend to basically brainstorm how we can solve this deadly malignancy. So this has been really an amazing thing that's worked really well. So John Bukvar is an amazing brain surgeon, and I work with a number of, he's one example I just want to highlight, one of the surgeons we work with, and he works on the Upper East Side at Lenox Hill Hospital, which is pictured here. And he and his colleagues are amazing. You can see in Newsweek and the best hospital, neurology, neurosurgery. Down here it says 2004, 2005, 2025. They're amazing surgeons, and they treat glioblastoma on a daily basis. They're looking the patients in the eye who have these really deadly tumors and doing their best. So they're great surgically and clinically. And here you see John also has this claim to fame with some other brain surgeon and some of his other colleagues on the Netflix series Lenox Hill. Has anybody seen it here? Lenox Hill? Okay, it's awesome if you haven't seen it. And what you might not know, even if you've seen it, they are not acting. They have cameras on their foreheads doing the surgeries they normally do. So you see their real struggles and everything they're doing. So check out Lenox Hill. It's quite amazing. For us, it's great because John comes to our lab meetings every week. Because of our collaborations, he's an adjunct professor at Cold Spring Harbor. So he comes every Wednesday when we have lab meeting. And it's really great because not only is he a great surgeon, but he has designed and carried out, I think, more than 40 clinical trials, many of which are for patients with glioblastoma. And he's a great guy. He couldn't be here today because he's with his kids at camp. He's the camp doctor in the Adirondacks. So it's an amazing team. I can't say enough about the team effort and how much that means with advancing the science. So I wanted to give you a glimpse of some of the things that our team is doing now. As I mentioned, these tumors they can be really large. I won't show you the nasty one again, I'll show you the schematic. They can be the size of a golf ball, even a baseball. So John and other surgeons remove these, and there's tons of leftover tissue after they're taking care of the patients. In fact, I don't know if anybody's interested, but here are some examples. Anybody want to touch them? That's actually a tumor taken out in some. It's all sealed. But you can pass it around if you want to pass it out. But it's very interesting. So this is tissue that normally would be discarded, and it normally would be thrown away. And so we study that in the lab. And what we can do is we can grow from individual patients, we can grow what we call these organoids. And this is actually a visual of some of these organoids in a dish that I recreated. But this is what they look like, these round spheres. And we think of these as brain tumor in a dish. And these are from individual patients. We put a lot of effort into making a collection of these that we can study. Because of course, what we want to do, we cannot do a lot of experiments in the people, obviously. So we use these organoids as a system where, in our case, we can target the heart of BRD8 and watch, for example, in a dish, watch those cells stop proliferating, and also transplant them into a real living brain of a mouse, and then give them, knock down BRD8. We have no drugs that target this really well. So in all these experiments, we're doing this genetically. But you'll meet Moses' team is here. Can you guys raise your hand, or do you want to raise your hand? Yeah, these guys are amazing. John Moses, I think, is traveling. The leader of the team, it says with John Moses. So that team is at Cold Spring Harbor. They're organic chemists. They have super cool technology called click chemistry, which I don't understand. They can probably tell you more. I think of it as Legos with chemical modifications. The point for me is they make hundreds of these compounds. And we can test them on ourselves in culture and all of our assays we've developed to see which ones take out BRD8. So this is an active program we're working on. We're really excited about this. In parallel, I didn't mention this, and we can talk about it if you need more detail, but in parallel, the bromodomain, the heart of BRD8, is really weird. There are other bromodomain-containing proteins, about 60 other bromodomains in this big family tree. But the bromodomain of BRD8 is an oddball. It's completely different functionally, and there's a lot of really interesting things we've discovered. But we don't know its structure. It has not been crystallized. So I don't think we have anybody from Leemore's lab, but Professor Leemore Joshua, sorry you can't see these names. She's down here. She is a crystallographer, world-renowned, and she figures out, basically makes what I think of as sugar crystals so they can get the three-dimensional structure, the beautiful structure, and even the atomic structure. So this represents, carbon, hydrogen, nitrogen bonds if you look closely enough. So that will help us design better drugs, and we're all collaborating on this. And of course, to try to figure out how BRD8 could be used. But the whole panel, this whole scheme could also be used for other personalized medicine for other targets. So the pipeline we've set up. So I want to acknowledge my team is phenomenal. I started by saying that, and I hope you get to meet some of the individuals tonight. And then we have great clinical neurosurgeons. I won't mention all the names. We have a lot of support, and in particular, we have had funding over the years for this project. And in particular, I wanted to acknowledge the patients, families, and foundations who have really, really supported and been really highly enthusiastic helping us advance this work. And for example, the Darlene Carbone Brain Tumor Foundation you should come if you're interested. You can come, each year we have this in May because that's Brain Tumor Month. And this is at Sunken Meadow State Park. It's I think normally on a Saturday. You should come. You get to meet the surgeons, the people that started the foundation, the families affected, and a lot of other advocates. So I strongly recommend this, and it's usually a really fun day. All right, thank you.